Okay, good afternoon, ladies and gentlemen. Thank you for coming back to our workshop series entitled Gastroenterology Research Opportunities in Antimicrobial Resistance, Metagenomic, Clinical Surveillance, and Wet Lab Experiment. It's a great honor to welcome our honorable speakers for today's session, Professor Wei-Chih Liao from National Taiwan University College of Medicine. I would like to introduce myself. My name is Langgeng Agung Waskito. I will be the master of ceremony and also the moderator for today's workshop. Recently, due to the global pandemics, university offices and other public spaces are being temporarily shut down. However, for students, lecturers, and other academicians, they are continuing their education through online learning and studying from home in order to improve their knowledge. The knowledge improvement is a crucial step in order to develop skills in many subjects for many professionals, including researchers. The pandemic situation enhanced many academic and research centers to provide online workshop and webinar in the interest of helping researchers to update the knowledge and informations. With this online workshop, the excellent speakers will share their updated knowledge regarding gastroenterology research opportunities in antimicrobial resistance, metagenomic, clinical surveillance, and wet lab experiments. Ladies and gentlemen, I would like to proceed to our main agenda for today for the International Workshop Series, Day 5, Section 2, with the theme of infection and cancers. For the first honorable speaker for today's session is Professor Wei-Chih Liao from National Taiwan University College of Medicine. Before we start, let me introduce him from his curriculum vitae. So he is a professor in National Taiwan University College of Medicine in Taipei, Taiwan from 2020 until present. His scopus achievements with the H index of 20 with the total number of publication is 63 with the total number of citation more than 2,000 citations. His academic background is a medical doctor from National Taiwan University College of Medicine, Master of Science in Epidemiology, Institute of Epidemiology and Preventive Medicine, National Taiwan University, and the last is PhD in Epidemiology, Institute of Epidemiology, and Preventive Medicine, National Taiwan University. His professional and research experience, the recent one is the professor in the National Taiwan University College of Medicine, and also Deputy Secretary General, the Digestive Endoscopy Society of Taiwan from 2016 until present. And his newest publication in 2020 is a deep learning to distinguish pancreatic cancer tissue from non-cancerous pancreatic tissue, a retrospective study with a cross-racial in external validations, and also relationship between pancreatic cancer-associated diabetes and cachexia. It is very interesting to see his CV. So without further information, so I would welcome to Professor Liao to present his presentation for today's session. Professor Liao, the time is yours. Thank you.
Okay, thank you. So thank you, Mr. Chairman, for the introduction. So can you hear me? Am I on the air now?
Yes, yes.
Okay.
Your voice is crystal clear. Okay.
Okay, so let me share my computer screen with you. Okay, so first I would like to thank the organizing committee for inviting me. And it's my great pleasure and honor to be here today. My topic is a practical of molecular and imaging approach for detecting pancreatic cancer in Asian population. And I have nothing to disclose. As all of you know, pancreatic cancer is increasing in incidence and mortality. And in the United States, it is projected to be the second leading cause of cancer deaths in the United States. So in Taiwan, pancreatic cancer now ranks 7th among all cancer deaths. And I'm not sure what is the rank in Indonesia, but I guess probably in Asian countries as in the Western countries, pancreatic cancer is rising. And pancreatic cancer has the lowest 5-year survival rate among all cancers, which is around 10%. So early diagnosis is key to improve the poor prognosis of pancreatic cancer. Why is the prognosis of pancreatic cancer so poor? The most important reason is that surgery is the only potential cure for pancreatic cancer, but most of the pancreatic cancer patients are diagnosed at a late stage when the tumor is already unresectable. Approximately 85% of the cases present with either locally advanced pancreatic cancer or metastatic pancreatic cancer. So roughly less than 15% of patients remain resectable at the diagnosis. And the study have shown that pancreatic cancer disseminates early, about 2 centimeters in size or greater. The tumor cell will gain entry into the circulation. Therefore, even if the patient seems to have only a tumor in the pancreas and undergo resection, most of the patients develop recurrence after surgery. So only about 2 to 3% of patients present with early pancreatic cancer, which by definition is less than 2 centimeters. And only in this very selected population that the possibility of a 5-year survival can be around 80%. It is important to know that early pancreatic cancer usually don't present with specific clinical symptoms. Therefore, it is very difficult to diagnose pancreatic cancer at this early stage. So how to increase the possibility of early diagnosis is a key issue and an unmet clinical need. So there are a few potential tools for early detection of pancreatic cancer. So the first kind belongs to circulating biomarkers. And the second kind of modality is imaging. So that is what I'm going to talk in the following part of my talk. So as all of you know, CA19-9 is currently the only widely used biomarker for pancreatic cancer. And it is approved for the follow-up of patients with confirmed pancreatic cancer. And the level of CA19-9 correlates with the poor prognosis. However, CA19-9 does not have a good enough sensitivity or specificity for the diagnosis or screening of pancreatic cancer. So its main use is for follow-up and prognosis prediction. However, a very recent paper published in Gastroenterology found that the level of CA19-9 increases exponentially since from 2 years before the diagnosis of pancreatic cancer, as you can see here. So when the specificity is set at 99%, the sensitivity of CA19-9 for testing blood collected 6 or 12 months before diagnosis, the sensitivity is 24%, which means CA19-9 can detect about 1/4 of pancreatic cancer. And when tested with blood samples collected within 6 months before diagnosis, the sensitivity of CA19-9 increases to 60%. So what this study tells us is that an increasing level of CA19-9 suggests that there is a high risk of pancreatic cancer. And the liquid biopsy is an emerging technique and an area of intense research. So liquid biopsy involves isolation of tumor-related materials from bodily fluid, which could include circulating tumor DNA or extracellular vesicles, or even the circulating tumor cells per se. So these tumor-derived products can be subjected to further analysis for making diagnosis or predict prognosis. So the concept of liquid biopsy has gained much acceptance because it is relatively non-invasive and it can be repeated multiple times over the course of treatment to inform about the disease progression and the treatment response. For example, you can see that here the cancer cells, as well as normal cells, can release cell-free DNA into the circulation. So we can isolate circulating DNA and use PCR-based methods to detect cancer-specific mutations in driver genes. So detecting these cancer cells in the circulation may facilitate early detection of a cancer. However, while this concept is very appealing, there are certain technical challenges. First, the circulating free DNA is low in abundance, and it is usually fragmented. So it requires advanced technologies to reliably detect these tumor-derived circulating DNA. So this is a seminal study of liquid biopsy in which the authors assessed the potential of liquid biopsy, combining detection of tumor-specific KRAS mutations in circulating DNA by a PCR-based method and protein biomarkers to detect pancreatic cancer. So as you can see here, the combination of KRAS mutation and the CA19-9 with a cutoff of 100 units per milliliter achieved 60% sensitivity for pancreatic cancer. Okay. So they further combined the circulating DNA KRAS mutation analysis and the CA19-9 with more additional protein markers, including CA, hepatocyte growth factor, osteopontin. And they found that combining all these assays can increase the sensitivity for pancreatic cancer to 64%. And the sensitivity for stage 1A is about 33%. And for stage 3B, 2B is 69, almost 70%. And an advantage of the circulating DNA assay is that it has almost perfect specificity. That is because the KRAS mutation is supposed to be occurring only in pancreatic cancer, not in normal cells. So in the cases tested in this study, only one of the normal healthy donor had a positive KRAS mutation detected. And because there is little overlap in circulating DNA positivity and the protein markers, so the authors found that there's little overlap between these two different technologies. So combining them together enhance the sensitivity for detection of pancreatic cancer without compromising its specificity. However, while these results seem very promising, there are a few caveats that should be considered. First, KRAS mutation is not specific for pancreatic cancer. It can also be seen in other cancers like lung cancer or colorectal cancer. So detecting KRAS mutation in the circulation does not necessarily mean that the tumor is in the pancreas. And the origin of cancer in patients who does not present an apparent tumor needs to be further defined. And the second is that these protein markers, such as CA19-9, can also be elevated in benign conditions. For example, CA19-9 is elevated in cases with cholangitis or cholestasis, etc. And in that study, these protein markers are tested using samples of healthy donors. So whether the results, the specificity observed in that study can be generalizable to a mix of patients with various cancers and benign conditions remain to be studied. Okay, and to address those issues, the same group of researchers, they further studied the possibility of combining KRAS mutation detection and also more genes, mutation of genes, and more protein markers to detect 8 common cancers. And the genes including known genes that serve as driver mutations in these cancers. And for example, you can see, we know that pancreatic cancer has 4 major driver mutations: the KRAS and the TP53 and the CDKN2A, cyclin dependent kinase N2A, and SMART4. And the 3 of the 4 driver mutations of pancreatic cancer is included in this panel. Okay. So by combining the driver mutation detection and the protein biomarkers, they were able to detect these 8 common cancers with a variable sensitivity. And for pancreatic cancer, the sensitivity is 70%, with a specificity of greater than 99%. And they further went on to try to predict the origin of the tumor by training a machine learning algorithm that uses information of the driver mutation status and the protein marker levels. And for pancreatic cancer, the trained model were able to correctly classify the patients as pancreatic cancer in 81% of the cases. However, There are a few things that must be remembered. First, in the test population, the cases of early or symptomatic pancreatic cancer, stage 1 cancer, is only 4%. So it is not clear if the sensitivity of the test remains high when it is used for screening pancreatic cancer with the aim of detecting resectable early pancreatic cancer. And again, the controls are healthy subjects. So let's move on to another tumor-derived material that can be used for liquid biopsy. It is the extracellular vesicles. The EVs are released by all cell types and can serve as a mechanism of waste removal or cell-to-cell communication. And importantly, in cases with cancer, EVs could serve as a way of establishing a pre-metastatic niche. The extracellular vesicle can be formed either by endosomes, can be classified as exosomes or microvesicles, and they are a source of cancer-specific materials with potential diagnostic and prognostic values. This is the first study that explores the utility of exosome for early detection of pancreatic cancer. And the researchers found that glypican-1, a proteoglycan, was enriched in circulating tumor-derived exosome. And you can see that here the box plot shows that in cases of pancreatic cancer, all the cases have elevated levels of glypican positive exosome, whereas the healthy controls did not have such phenomenon. So there's a clear separation between the normal and the pancreatic cancer cases. So they found that glypican-1 positive circulating exosomes detected in serum could differentiate pancreatic cancer patients with absolute sensitivity and specificity. And then they further showed that such exosomes carry KRAS mutations that has the same mutation in penins in animal models, suggesting that these extracellular vesicles are derived from the tumor in the pancreas. While this study shows a very promising result, supporting that such a glypican-1 positive exosome could perfectly differentiate pancreatic cancer from controls, later studies could not reproduce the finding. And then another group of researchers, they try to also use the isolation of exosomal proteins for liquid biopsy to detect pancreatic cancer. And they isolate the exosomes from cancer cell lines of pancreatic cancer. And they found that the 6 surface proteins are specifically expressed in pancreatic cancer-derived exosomes. So they developed an immunocapture assay using the 6 surface proteins to enrich cancer-derived exosomes. and then subject these exosomes to subsequent molecular profiling. And you can see that with this immunocapture assay to enrich the exosomes, the sensitivity of detecting pancreatic cancer using a PCR-based KRAS mutation detection method, the sensitivities are increased compared with the sensitivities obtained without enrichment. So in cases with metastatic pancreatic cancer, almost 80% of the cases can be detected using this assay. And then we move on to another way of liquid biopsy, which is to use circulating tumor cells. As shown in this diagram, tumor cells can gain entry into the circulation. And by detecting circulating tumor cells, it may facilitate early detection of pancreatic cancer. However, circulating tumor cancer cells are low in abundance. And approximately one circulating cancer cell is accompanied by 1 million white blood cells and 1 billion red blood cells in a milliliter of peripheral blood. Therefore, isolation of circulating tumor cells is very challenging. And so far, the most widely used and approved method for isolating circulating tumor cells is the cell search technology, which uses an epithelial cell adhesion molecule, a surface protein, which is frequently seen in cancer cells, to select the circulating cancer cell. So using this way, various studies have attempted to diagnose or detect early pancreatic cancer with the sensitivities ranging from 20% to 64%. So it's quite variable in the literature. Overall, the sensitivity for pancreatic cancer detection remains modest. And an important reason is that it depends heavily on the isolation and the detection technique used for isolating the circulating tumor cells. And also, early stage pancreatic cancers shed a small number of circulating tumor cells, therefore are more difficult to detect. Sorry for the interruption. So move on. So I would like to introduce a novel way of liquid biopsy. Is to use the 5-hydroxymethylcytosine signature. And this is a recently discovered epigenetic modification of DNA. And it is linked to gene regulation and has been shown to play important roles in cancer pathogenesis. As all of you know, the cytosine in DNA can be methylated at the 5th carbon. And an enzyme called TET methylcytosine dioxygenase can reduce this methyl group into a hydroxy group. And this 5-hydroxymethylcytosine, 5-HMC, this can serve as a signature. So in this study, they found that by detecting 5-hydroxymethylcytosine signature in cell-free DNA, they can inform about the presence as well as the type of tumor. So in this study, they conducted whole genome cell-free 5-HMC sequencing of the peripheral blood. And they found that different cancers can be classified based on the profiles of the hydroxymethylome in the cell-free DNA. So this is a very recent paper published in Nature Communications. And the researchers further assessed whether by profiling the 5-hydroxymethylcytosine signature in circulating cell-free DNA, whether it can detect pancreatic cancer. And they found that compared with healthy controls, pancreatic cancer patients have very different 5-hydroxymethylcytosine signatures. For example, in the promoter regions, pancreatic cancer patients tend to have a lower 5-HMC density, whereas in the 3 UTR region, pancreatic cancer patients tend to have increased 5-HMC density. So they profiled the whole genome, and then they found that 577 genes and 217 genes have increased and decreased 5-HMC densities in pancreatic cancer compared with healthy controls. And they found that the genes with increased 5-HMC densities tend to be upregulated in cancers with KRAS or P53 mutations. So this provides further support that the 5-MHC signature can inform us about the presence of cancer and its genomic profiles. And they further went on to use select meaningful genes to construct a predictive model. And they found that this model could classify patients with pancreatic cancer. And you can see that the blue dots are pancreatic cancer patients that can be detected with the CA19-9. And the red dot, sorry. And for with this prediction model, they could differentiate between pancreatic cancer and the control, achieving an AUC of 0.92. And compared with the CA19-9, this prediction model were able to predict more pancreatic cancer, even cases with stage 1 pancreatic cancer. In all the pancreatic cancer patients, only one case were missed by this predictive model. So the result of this study supports that the 5-HMC signature may be used to facilitate early detection of pancreatic cancer. Okay. So the bottom line is that circulating free DNA hydroxymethylome in pancreatic cancer patients are differentially enriched for genes commonly deregulated in pancreatic cancer. And the models based on 5-MHC density differentiated between pancreatic cancer and controls with an excellent AUC in independent test sets. And this change can be seen even in early stage pancreatic cancer. So so far what I have talked about is to use liquid biopsy for screening asymptomatic patients. And there are other novel markers that may be used to detect patients with subtle symptoms of pancreatic cancer. For example, recent studies have shown that pancreatic cancer-associated diabetes is a paraneoplastic syndrome, which occurs in about 40% of pancreatic cancer patients. And this special form of diabetes is characterized by new onset diabetes within 2 years preceding the diagnosis of pancreatic cancer. And it is induced by unclear tumor-derived factors to induce insulin resistance. And the previous studies have shown that at the onset of pancreatic cancer-associated diabetes, the cancer is usually resectable. Therefore, the pancreatic cancer-associated diabetes provides a good window of opportunity for detecting pancreatic cancer at an early resectable stage. And this kind of diabetes is characterized by progressive hyperglycemia with weight loss and a marked insulin resistance. However, the mechanism and the mediator of insulin resistance is not quite clear. However, if we want to use this as a way of detecting early pancreatic cancer, we face the problem that diabetes is so prevalent. And the previous studies have shown that in new onset diabetes after 50 years old, only about 1% of the cases can be attributed to pancreatic cancer. So how can we find those cases with pancreatic cancer associated diabetes from the so many patients with diabetes? Therefore, we think we need to identify the factor that mediates the insulin resistance. And if we can measure this factor, we might be able to differentiate pancreatic cancer-associated diabetes from type 2 diabetes. Therefore, we conducted a study to identify the diabetogenic factors produced by pancreatic cancer. And we assess whether these diabetogenic factors could help us distinguish pancreatic cancer-associated diabetes from type 2 diabetes. So we started from profiling the conditioned medium of pancreatic cancer cell line, and we used the proteomic profiling together with analysis of genomic data. Then we found that 86 proteins are differentially secreted and upregulated by pancreatic cancer cells. And then we use bioinformatics analysis to search for potential candidates that may mediate insulin resistance. Then we selected a few candidates, and then we went on to test if they could mediate insulin resistance. And we found that S100A9 and galectin-3 can dose-dependently inhibit insulin-stimulated glucose uptake in muscle cells, suggesting that these two factors that are produced by pancreatic cancer cell lines can induce insulin resistance. And these two factors are pro-inflammatory factors, and they have been shown to promote pancreatic cancer proliferation and migration. And acid by S100A9 forms heterodimers with S100A8 and can bind the toll-like receptor 4 and amplifies inflammatory response. So we further examined whether these two factors are overexpressed in pancreatic cancer-associated diabetes patients. And we found that These two factors have a weak expression in tumors without pancreatic cancer-associated diabetes, but are strongly expressed in tumors inducing pancreatic cancer-associated diabetes. And then we evaluate their potential for differentiating between pancreatic cancer-induced diabetes and type 2 diabetes patients. And we found that at the chosen cutoff, S189 could achieve a sensitivity of 70% and a specificity of 74%. And measuring galectin-3 at this cutoff could achieve a sensitivity of 62% and 80% specificity. Then we further checked whether these two markers are sensitive for early pancreatic cancer. And we found that even in stage 1 pancreatic cancer, these two markers achieved a sensitivity above 70%. So this is the first study that identified the diabetogenic factors produced by pancreatic cancer that mediate insulin resistance in pancreatic cancer-associated diabetes. And we further showed that these two biomarkers could facilitate differentiation between pancreatic cancer-associated diabetes and type 2 diabetes. Okay, so far I've talked about the biomarker and the liquid biopsy for early detection. So the next part I would like to move on to the imaging part. As all of you know, we have several modalities for detecting pancreatic cancer. The most commonly used is CT, computed tomography. And we also have magnetic resonance imaging, MRI. And furthermore, we have endoscopic ultrasound, which is an endoscopic procedure that has the highest sensitivity among all modalities for pancreatic cancer. As you can see here on this CT image, there is a hypo-intense tumor at the head of the pancreas and with a dilated pancreatic duct. And this is very typical for pancreatic cancer and very easy to diagnose. However, that is not always the case. For example, on the right panel, this is a case who presented with obstructive jaundice with a dilated bile duct and a pancreatic duct, suggesting the possibility of a pancreatic head tumor. However, there is no clear tumor on CT at the pancreatic head. So because of the high clinical suspicion, the patient underwent the EUS, and there was a 1 centimeter faint hypodense tumor at the head of the pancreas. And a EUS FNA proved that this tumor is a stage 1 pancreatic cancer. So this case highlights the difficulty of detecting early pancreatic cancer on CT. In fact, previous studies have shown that the sensitivity of CT for pancreatic cancer smaller than 2 centimeters is only around 60%. That means that almost 40% of pancreatic cancers less than 2 centimeters in diameter could be missed on CT. And this is further compounded by disparities in radiologist experience and expertise. Although we can use EUS to confirm whether a tumor is present or not in cases with suspicion, EUS is an invasive procedure and it requires technique, it requires experts and dedicated US equipment. So it is not widely available. So we cannot subject all cases to US. So in real clinical practice, it is very often to see patients with symptoms suggestive of pancreatic cancer, but the CT did not show any clear tumor. So very often, the patients do not receive further evaluation. And because of the rapid course of pancreatic cancer, very often the patient will become clinically apparent within a short period of time. But at that time, the pancreatic cancer very likely becomes unresectable. So to tackle this limitation, I think computer-aided diagnosis detection tool is needed to improve the sensitivity for small pancreatic cancer. Given the recent advances in artificial intelligence, we asked whether AI could help us solve this issue. As all of you know, AI has become a new player in medical diagnosis, and the FDA has already approved an AI-powered diagnostic tool for diabetic retinopathy. So this is attributed to the recent advances in deep learning, a branch of artificial intelligence. Specifically, a computer algorithm called convolutional neural network has shown great promise in image analysis. So we tried to assess whether the CNN could help us differentiate between pancreatic cancer and normal pancreas. So this paper was published last year in the Lancet Digital Health. So in this study, we used the pancreatic cancer and the control subject identified from our hospital, and we retrieved the images for training a CNN for differentiating between pancreatic cancer and normal pancreas. And then we tested this algorithm in our test set, as well as an external data set consisting of pancreatic cancer and normal pancreas images from the United States. So when we conducted this study, there were two approaches that we tried. The first is to analyze the pancreas as a whole, like a panoramic view. But we found that it is not working, probably because the cancer can be very small, and most of the pancreas outside of the tumor is relatively normal. So it is difficult to train a model to specifically recognize the relatively small tumor in the pancreas. So instead, we used an analysis by patch method. So we cut the pancreas into small patches so that we look at each patch individually and see if we could spot any suspicious area in the patch. So this shows the concept of our methods. So we manually segmented the pancreas and cut the pancreas into patches. And the patches containing pancreas tumor is labeled as cancerous patch. and the patches containing only normal pancreas is labeled as non-cancerous patch. And then we train a CNN to distinguish cancerous patches from non-cancerous patches. And then we determine whether a patient has pancreatic cancer or not, based on the proportion of patches that are predicted as cancer. So the pancreas is first manually segmented as well as the tumor. And then we have to normalize and crop the pancreas and tumor into patches. So as I just have said, that this is the flow of training and testing. So the results, the model that we trained has 3 main convolution blocks. So in the local test set 1, the patch-based analysis, which means to distinguish patches of cancer with patches of normal pancreas, achieved a sensitivity of greater than 90% with a specificity of around 86%. And more importantly, and the similar results were also noted in local test 2, another test set with smaller tumors from our hospital. And more importantly, is the patient-based analysis, which determines whether a patient has pancreatic cancer or not. And in local test set 1, the CNN achieved its accuracy of almost 99%, with the area under the ROC curve of almost nearly 1. And most importantly, you can see that the sensitivity of a radiologist in our hospital for these test images is 94%, which is slightly lower than 97% achieved with the CNN. And in local test set 2, we can see the similar trend, with the sensitivity of the CNN significantly higher than the sensitivity of a radiologist. So we found that the trained CNN achieved higher sensitivity than radiologist. And among the 12 cases missed by radiologists, the CNN were able to identify 11 or 92% of the pancreatic cancer. So we think this model could supplement the radiologist for detecting early pancreatic cancer. These 2 cases are cancers missed by radiologists. On the left panel is a patient presented with acute pancreatitis. And later, the patient was found to have pancreatic cancer. And in retrospect, we found that there is an early pancreatic cancer hidden here. And on the right panel, this patient presented with a dilated bile duct, but there is no apparent tumor in the pancreatic head. And this patient was later confirmed to have a stage 1 pancreatic cancer, about the size of 1 centimeter here. This is proved by surgery, resection of the tumor. So these 2 cases are correctly classified as having pancreatic cancer by the trained CNN model. We also checked the sensitivity of the model according to the size of the tumor. And we can see that for tumors less than 2 centimeters, our radiologist achieved the sensitivity of almost 90%, which is very high compared with the previous studies. Probably because we have a lot of pancreatic cancer patients in our hospital. So our radiologists are very familiar with the manifestations of early pancreatic cancer. And the CNN also achieved a sensitivity of 92%. And this shows the real location of the cancer and the suspected location of the tumor by the CNN model. You can see that the suspected tumor by CNN corresponded with the real tumor. So I would like to wrap up my talk. Firstly, current liquid biopsy technologies still have limited clinical acceptance. This is because it is time and labor consuming and potentially costly. And there are important technical challenges that still needs to be resolved. For example, isolation of cancer-specific exosomes are quite challenging. And the circulating DNA and the circulating tumor cells are low in abundance. However, liquid biopsy does present a viable potential solution for non-invasive detection of early pancreatic cancer. Secondly, the imaging detection of pancreatic cancer is limited by the inadequate sensitivity for small pancreatic cancer, and AI might supplement radiologists for early detection. However, the liquid biopsy methods and the AI methodologies needs to be further verified before they could widely applicated in general clinical practice. They need to be evaluated for their robustness and their generalizability to the real clinical setting. And the applicable settings for them to apply needs to be defined. And their potential benefit or impact needs to be further assessed. Thank you for your attention.
Okay, thank you very much, Professor Liao, for a very great presentations about the new approach for both imaging and molecular detecting the early cases of pancreatic cancer. So yeah, again, pancreatic cancer seems to be very low survival rate. As we know, the very famous one, Steve Jobs, was also died from the pancreatic cancer. So it was like previously already have a surgery and said okay, but later in the 2014, then he was died due to the complication of the pancreatic cancer. It is very interesting. And again, in every cancer in the world, also the early detection is very necessary to reduce the mortality rate. Okay, so for everyone who has any questions for Professor Liao's presentation, you can drop your questions over the Q&A button below, and we will still gathering some questions, and the question will be discussed after the second speakers from the Dr. Firman Setiawan. Okay, so for everyone, still stay tuned for this workshop. So then we are going to move forward to the second speaker, from the Dr. Firman Setiawan. Okay, so I'm going to show his resume. Okay, excuse me, Professor Liao, could you please, okay, stop the share screen. Okay, I'm going to show the CV for the Dr. Firman Setiawan. Okay, so he is the PhD student in Juntendo University, Graduate School of Medicine, Department of Neurology, and from the Division of Stroke in Tokyo, Japan. And then his training is antibiotic associated therapy from Erasmus Medical Center, Netherlands, and also the Carbapenem resistance Enterobacteriaceae from Kobe University, Japan. And his research publication is the prevalence of methicillin-resistant Staphylococcus aureus from nose and throat of patients on admission to medical wards of Dr. Sutomo Hospital, Surabaya. And he is also currently becoming a staff in the Department of Microbiology in Faculty of Medicine, Universitas Airlangga. All right. So without any hesitation, so I would like to welcome to Dr. Firman Setiawan to
Okay.
share your screen and
I share my screen. Okay, thank you for
yes,
Dr. Langgang. I share the screen. You can find it. Not yet.
not yet.
View screen. Share screen, right?
Yes, yes, yes.
Share screen. Enable control and application. Yes. Okay. You found this?
I'm sorry, I still not yet to
Not yet.
see your screen.
Share on Windows and application. Thanks. You find it? Not yet. Okay.
Okay, so we still cannot see your screen. Uh, so if possible, could you please send us
Okay.
your presentation so the screen will be shared from here? You can send me. You can send me now. Okay. So excuse me for all the participants for today's workshop. We have some technical issues, so we will back later for a while. Okay, so thank you for the patience. So I'm going to share the screen for the presentation. Okay. Okay, Dr. Firman, so you can start.
Okay, thank you, Dr. Langgeng, and I'm sorry for little trouble from this moment. Okay, Assalamualaikum warahmatullahi wabarakatuh. Konnichiwa minasang. Hello everyone. I am Firman Setiawan. I am clinical microbiology specialist medical doctor in Erlangga University, and I know also PhD student from the research group for infection control science at Chunten University until this month. And next month, I changing to neurology department to study about gut microbiome modulation to brain ischemia therapy. Okay, we start about molecular epidemiology and phylogenetic analysis of antimicrobial resistance using NGS or next generation sequencing. So we know that antimicrobial resistance or AMR is increasing threat to public health and sustainable development, and also in pandemic era like now. Uh, in September 2020, 2020, WHO with the Global Action Plan on AMR surveillance to strengthen the knowledge and evidence base for informing strategy and monitoring the effectiveness of intervention. And what they choose, they choose whole genome sequencing to surveil antimicrobial resistance. What is the whole genome sequencing? Whole genome sequencing, or WGS, provide a vast amount of information and the highest possible resolution for pathogen subtyping. Uh, the application of WGS for global surveillance can provide information on the early emergence and spread of AMR, and further inform to policy development on AMR control. Also in Indonesia, AMR control is needed to do by government. This is different with the traditional phenotypic typing system based on the microbial or biochemical, serological, or physiological character of the pathogen. So have been widely used for the past. So sequencing data from AMR surveillance may provide key information to guide the development of rapid diagnostic tools for better and more rapid characterization of AMR, and this complement phenotypic method. So how to perform NGS for AMR prevention and control? So before we discuss and actually starting doing our analysis on next generation sequencing, I would like to give you introduction about the roles for the microbial typing. But before that, is better we adjust in same mindset about terminology in infection control definition. So the important definition is, first we should know about the epidemiological typing. What is epidemiological typing? Epidemiological typing is this is a term in common use. However, it is not to be encouraged, but because to isolate are phenotypically or genotypically indistinguishable, does not mean that they are related epidemiologically. This has to be proven by epidemiological investigation performing describes data analysis. Uh, Preferred term will be typing to explore epidemiological hypothesis. Like there is a little point using typing as an fishing tools. Uh, and there has to be an epidemiological focus and appropriate to collection of organisms submitted for typing, criteria referring for the isolated to laboratory to enable proper interpretation of the typing result and the likelihood that typing can provide a helpful answer to the problems posed. It is apparent in much of the literature and that the reason for the typing have not been tough much time and effort and resource have been spent with little to show that can stand up to scientific. So this is important terminology in infection control uh, uh, and uh, science. So the second terminology we should know is about the microbial epidemiology. What is the microbial epidemiology? The microbial epidemiology is the study of the distribution and dissemination of microbial pathogen, including their transmission pattern, risk factor for infection, and preventive and control measure of the infectious disease. And also, what is the microbial strain? Microbial strain is an isolate of a group of isolate that can be distinguished from the other isolate of the same genus or species by phenotypic and or genotypic characteristic. When sampling for typing, it is wise to pick 5 distinct colonies, like we pick 5 colonies in one plate to reduce the chance of missing of the mixed strain. So the strain is important because we can distinguish between others. And the third is about clone. What is clone? Clone is a bacterial culture isolate independently from different source in different location and perhaps at different times, but still showing so many identical phenotypic and genotypic trait that the most likely explanation for this identity is a common origin. So we can identification the common of origin of the pathogen. And also, what is the clonal variant? And the clonal variant is the recent or the recent or descendant of a successful clone which have undergone minor evolution changing. So one clone can't have a variant. When we using genome analysis, we can distinguish this variant. And also, what is the clonal complex or group? Complex or clonal group is bacterial isolates showing a high degree of similarity. Ideally, based on near identity of multi-locus enzyme profile and multi-locus sequence type identically to a clonal group. So, and this the last we should know is clade. What is clade? We rare to hear it. This is evol- evolution of term that is appearing increasingly in the infection prevention and control or hospital hygiene and also in antimicrobial stewardship literature. Uh, this pathogen where have a DNA sequencing has been performed, it perfect to grouping of microbial isolates, even those that are extinct descendant from a common ancestor. Okay, next slide, please. Next slide. Okay, so why we should perform whole genome sequencing or uh, whole genome sequencing, yeah, because we cannot manage what we cannot measure. So make sure you can measure it. How to measure it? The answer is microbial typing. So what is microbial typing? Uh, the roles of microbial typing for infection control science and AMR, uh, reducing the AMR spreading, this is essential part and important role for the infection control. Role for the microbial typing in uh, infection control, prevent, and antimicrobial stewardship is important. First of all, microbial typing is essential part of the function in supporting healthcare associated infection, and also can play an important role in informing and investigating of AMR spreading. Uh, there are many reasons why microbial typing might be considered. This can be considered under the following broad titles. In Japan, clinical laboratories will vary in what they can offer, but they may also submit isolated to a larger central or reference laboratory, depending on easy to access resource and ability of the service to supply useful typing information is in a timely manner. So in Japan, we have a good system for uh, this issue. First, uh, why the important and essential? First, incident or outbreak investigation, local, national, and global. Global. Why we doing this one? Because now in the world, this easy. Some people migrate to another place, and so easy to uh, the pathogen or AMR can spreading around the world. So this is will include the examination of microbial isolate from epidemiologically related cluster of patient and staff infected over varying period of time. So we can uh, knowing what time they get the infection or colonization. It might also be employed to investigate what are thought to be endemic or sporadic cases. It is essential that clinical infection control team members consider to the hypothesis that need to be tested. So if we suggest some cases, we can doing perform this measure. This should be informed to the cycle of infection. Infection cycle infection should be break it. So before we break it, we should know how they spreading and transmission. Which part of the microbial reservoir uh, before? Okay, and the second, what is the route of transmission of the strain, and what are the pattern of their spread? Although the index case may be transmitted in particular way, such as in the operating theater during surgery, subsequent route may be, for example, via unwashed healthcare worker hands or the word. So this is important. And then number three, and the third, what are the port of entry into the patient? This may be, for example, via wound, injection, cannula site, airborne, or oral portal. So after we know the portal infection, we can cut or break the chain of infection. And the last, number 4, and the fourth, as the outbreak spread, so one may need to identify the enlarged reservoir, for example, patient care in the many outbreaks of pathogen or hospital infection outbreak. 70% or more of affected individual are colonized rather than infected, either of which can be source of further spread. So it means that if we 70% of the highest cases, hospital or nosocomial cases, because the pathogen is colonized in the near of the patient. So next slide, please. So interacting of IMR surveillance or nosocomial surveillance data with microbial typing information is performed increasing and is of more use if there are globally agreed method and standard and database of strain to refer to. And in this way, uh, we should link between microbial type and spread, how to spread they spreading, and severity of the infection caused by certain clones and association with specific uh, for example in the toxic shock can be described and prevention and control uh, to monitored in this case. And for the criteria, perform criteria of typing method. So criteria for assessing microbial typing system. For example, a criteria can vary depending on the level at which typing is being used. Like locally at the healthcare institution, like in the hospital or long-term care facility. Like in Japan, we have uh, or in Indonesia, we can use it in Lansia ward. And and also we can use locally, regionally, and globally the data. Like locally, we can and regionally we can take the reference laboratory and research center. Like in Erlang University, maybe we have a research center about this study. And globally, increasing there are no dedicated network and internet accessibility database. It means that if we go to another country, we should know what happened in the uh, country we want to do, like in the COVID-19 pandemic. Okay, next slide. So for the goal of AMR surveillance, first to monitor trend for the local prescribing guidance to, yeah, we can read this one. This is common, maybe from the AMR we can hear. For the next slide, please. Define what is you wish to monitor. So when we doing molecular typing or microbial typing, we can what we want to uh, take it, like trend of resistance. This is decided on organism or specimen type, antimicrobial resistant pattern to be monitored, or new mechanism of resistance, like impact of new mechanism of resistance because the um, microorganism easily to mutation. So maybe they have a new mechanism. So why we using next generation sequencing? And the last impact of the other activities, such as reducing the Clostridium difficile infection uh, with altered prescribing with potential to select different antimicrobial resistant. That is important. So PPRA in Indonesia. This is workflow of how to whole genome sequencing doing in clinical laboratory. Yeah, maybe clinical laboratory or microbial laboratory. I hope we can you can understood after I explain it. Okay, stay tuned. Next. So uh, for for the understanding about how to doing the whole genome sequencing, in my experience, I doing by MySec workflow. So how to do that? I would like to give you introduction about next generation sequencing or NGS or whole genome sequencing using NGS. I repeat it. Uh, and right now, the sequencing technology can be divided into next generation sequencing and third generation sequencing. What is difference? The difference between them, this next generation sequencing, you have to amplify your fragment before you start sequencing. But the third generation sequencing, you can actually sequencing directly to the single molecule, or you can call third generation sequencing at the single molecule sequencing. So third generation sequencing is similar, or you can uh, uh, call the single molecule sequencing. And this is the machine or sequencing platform that you can use in each of the technology. But I would like to focus only the next generation sequencing for today. So this is an overview how to you can doing next generation sequencing machine. Okay. First of all, uh, you have to do library preparation. So um, in the right side, we can doing a library preparation. This is a special step in the MySec workflow. You have to do library preparation, and the first step of the library preparation, as I told to you, amplify fragment sequencing. So and the first step, you have uh, to fragment your DNA. So we extract the DNA. After that, you fragment it. You fragment it to be uh, a short chain, and you ligate your fragment DNA with adapter. So next slide, please. I can next. This one. And uh, you after you fragment the DNA, then you ligate your fragment DNA with adapter. And what is actually inside the adapter? Adapter is contain amplification primer that used for amplify the fragment, and then it contains sequencing primer for the sequencing the fragment. So it's mean that after we run the machine, you have more than one sample. So it means that this is fast, not only one by one, but you can run more than one sample. you have to put barcode, and the barcode is same like a primer. The barcode is sequence that's specific for each of the samples. So it allows you to put more sample in one sequencing run after you locate the adapter to your fragment, and then you can start amplify your fragment and then put uh, those fragments into the sequencing machine and start sequencing. Before we actually could code and see what the data you will get from the sequencer, let me I tell you a little bit briefly about what the standard format of the DNA information. Okay, next. Uh, yeah, before this one, I will tell to you a little bit briefly about what the standard format to store DNA information. The standard format to store DNA information we call FASTA. Next slide, please. This is um, uh, technically how to machine doing our experiment, right? Uh, okay, we back in the format store of DNA information. So after we finish, okay, next, next. This is technically because every laboratory may be different, a little different procedure. Okay, this is analyze data. After we finish the standard format of store data information, we call FASTA files. So after we can get from the machine the FASTA files. In the FASTA file, it contains two lines. The first line is header then start from, like sign like in this, and then you can put any ID. Please before a slide before uh, before, right? Oh, correct. Uh, the second line we have two uh, line, and the second line is sequence data, and you can it can be in one line or multiple line, and in one FASTA format you can put more than one sequence, and you can start another sequence by starting a new line with uh, I mean that you can put ID and sequence and so on. This is the standard format for the store sequencing data. They can call FASTA, right? But it's not the format that you will get directly from the sequencer. What can you get from the sequencer? So we can have the DNA, our fragment DNA doing library preparation. You sequence the DNA, and what you get, you are going to get a source sequence that we call read. Next, after the slide after. Okay, next. This one. So we get the source sequence that we can read, or we can call raw reads, and this is what you are going to get from the sequence machine by NGS technology. So the raw reads, what you will get is not going to be in FASTA format, but it's going to be stored in FASTA FASTA format using Hue FASTA format. So what is FASTA? Is actually FASTA or FASTA plus quality scores. So instead uh, of having two lines like in FASTA, you are going to have four lines for one read or one sequence. So what four lines. So the first line, of course, is the header or the ID of the read, and the second line is this DNA sequence, and the third line is the optional lines that you can fill in whatever. And the last line is the quality score. Why it's called FASTAC, because it's contain DNA information and the quality scores. Sequencing quality scores, it means that sequencing quality scores. So FASTA contain 2 lines, while FASTA contain 4 lines with the quality score. That is the format that you will get the from sequencing machine. Okay, the next. After we get this one, uh, okay, after we get the format data from the machine, we can. I will show you. This is my job before. I doing work this one. Uh, we can sequence one direction that what you can single and read, and you can sequence two direct that we call pair and read. So in this, what we just uploaded into the website, using Aura server. So because they have a big data, they can uh, measure our sequencing data in the database, big database. Okay. Uh, and when you do paired and read, what you can have fragment gap. Uh, before slide before. Ah, so we can get the pair gap. What is no no? We can have the fragment gap, and we are going to have another term for pair and read that we can call insert size. So we can insert the size of in the size of the read together with the fragment gap, not included the adapter. So after that, they can build in the machine, in the uh, database, not include the adapter. So again, insert the size mean all the fragment not include the adapter. So that read, they read and the fragment gap, that the insert the size and the insert size vary according to the sequence technology. Some procedure very small insert size, some procedure very large insert size. So this variation, now you know what the data look like from the sequencing machine. So what you are going to do the next for the data or raw reads. Next. So after, uh, yeah. So how to doing the data analysis from the whole genome sequencing? So first, uh, because like I told to you, you normally have more than one sample into the run. So in input sample contain barcodes that specific for example. So first of all, you choose properly barcode or split the barcode from your raw reads, but this is normally. It has been done by the sequencing machine, and the next step is to trim the read, because you have some read with low quality, or some read contain uh, un uh, unreading. Uh, so we trim the read or the part of the read that contain low quality DNA, or contain something and like this. Next slide. This is how to data analysis we using online or web-based um, we're using a web based. Okay, the next slide. This after we inside in the web, we can get the reply of the email from them, and easily we get what's mean result of the our data. So the coverage is an average number of times of the road read that cover in your genome. Remember, you amplify your raw data, your the I mean the fragment before sequencing than you. If your raw reads have been sequenced with good quality, you are going to have high coverage. So, and when we doing what is mean data analysis of the whole genome sequencing data, so we can uh, get the good result. So how can you calculate the coverage? If you know number of reads, the read length and the genome size, you can know the coverage. For example, in my work before, my row reads contain 5 million base pair. But the length of the read is just only 10 base pair. So 5 million reads and just 10 base pair. We just read the uh, 100 base pair. So this is need much of uh, length of the reads, and the genome size in the genome is more than 5 megabase pair. So in this case, the coverage is fold 100-fold coverage. The meaning that the average 100 read cover each position in our genome in this quality in this quite very high. Another parameter is similar to coverage, but we can calculate of the coverage. So if you divide it, next slide, please. Okay, if you divide it by the assembly size and the target size, so I will tell you more about assembly. Uh, the thing is, you have raw reads and you try to assemble them into something that we call contig. Okay, the raw data fragment and we assembling into contig in the website, and then we can sum up all of database pair in our assembly, and then you will know the assembly size. For example, when I do it assembly for the MURSA genome, I got 3 million base pair, but my target genomes is just only 2.5 million base pair. In this case, the breadth of coverage is only 0.8, nearly 1. One is mean that one is it mean that the perfect cases, meaning that you sequence everything uh, of our of my genomes. And another parameter that we can call in depth. So the next okay, this is my work before. This is a MURSA strain from the top referral patient, top referral hospital patient. So this is get the peak base pair of the whole genome sequencing, and we can get easily uh, analysis whole genome sequencing not only the strain but also the uh, virulence chain, and also we can compare with another strain around the world. Okay, so first of all, we can do the novo assembly because the raw rate is very short. So we can try merge them to make a longer sequencing, and then after that we can analyze in this website. Okay, after that, um, if our chain map the read to the reference genome, like I using uh, Mursa and uh, 2013, right? And my strain is as a MRSA number 18. It means that what the we normally do it for the identify or detect any point mutation like SNP, for example, in order to that thing as some to assembling or mapping the read to the reference genome, and we don't normally cannot do it by hand, but by a machine and by computer. Of course, we can do it using a program or software, and when we compare, we can get the cap white line in the genome, and most of the software or tools for doing assembly or mapping of the reference genome normally, most of them they are in uh, command line, but when you're using uh, Windows or Mac, we can buy the uh, software and install in our computer. So uh, we scientists, microbiologists, or we can anyone can that have any skill to using computer command line in the command line. So I think the better we can buy and the software easily. Next slide. Yeah, and also we not only can um, analyze the uh, chromosomal chain, we can also find the plasmid base. Uh, what is plasmid base for the IMR mechanism? For the example, if we doing like this one, uh, in MRSA, no, no, in the plasmid finder, we can easily found in the gram negative, because in the majority case of the gram negative, isolate detected by uh, what is it? A plasmid. Next slide, please. So, for example, um, in the majority of cases, next slide. Uh, this easily we can search the plasmid finder in the our data. So, on the next slide, can you see? I will show you for the yes, this is uh, we can when we submit the data and we sort the plasmid, we can easily to get this data. So the next for the example, this is how to implementation uh, whole genome sequencing in infection control in hospital using to detect plasmid mediated outbreak. So in our cases, in the Juntendo, we have um, uh, 6 hospitals, so they can we can easily to knowing the outbreak. So next, next slide. Ah, this is plasmid mediated outbreak. So in the majority uh, cases of the first plasmid mediated outbreak detected by phenotypic identification. Like in this case, we can know in the uh, begin the outbreak, we can found the Klebsiella pneumoniae one by one, following the time, and also we can uh, get the another uh, Klebsiella pneumoniae like Escherichia coli, and then the Citrobacter froundi in the outbreak. So when we uh, detect the plasmid mediated, we can what does it mean? The species. Oh no no. Uh, in the majority cases, in this plasmid mediated outbreak, it means that the first CRI or carbapenem resistant Enterobacteriaceae, we detect phenotypic Klebsiella pneumoniae, but not only the Klebsiella pneumoniae, we can follow by Escherichia coli. Like in the case, we can found Citrobacter froundi and Enterobacter aerogenes pathogen in one case. And of this first case of CRA, in these cases, right, uh, were detected into phases, and the result of screening for the carriers, while the remaining about isolated were recovered from the virus clinical sample, including urine, wound, aspirate, drainage fluid, blood, or respiratory tract samples. And this is uh, cases, we can look in figure B. You get it. One large cluster of the isolated of the Klebsiella pneumoniae, in this case is ST392 and 30 core genome SNP from the root to tips carrying the plasmid with the uh, KPC gene. And in this case is and in additional isolate carried the plasmid, but this is did not belong to the Klebsiella pneumoniae, the same cluster, but we also can found it from the Escherichia coli and the Klebsiella pneumoniae and the sequence type other than ST370 2 and 3 were another uh, pathogen like a Citrobacter spp. um, there was evidence of both clonal expansion of the Klebsiella pneumoniae strain and the plasmid spread to other sequence type or species. It means that the plasmid spreading not only just one uh, uh, not only just one in the Klebsiella pneumoniae only, but they can also spread to other species like a Citrobacter and like a Escherichia coli, like Klebsiella pneumoniae and others. So in gram-negative, the plasmid mediated uh, outbreak, we should measure if you want to uh, detect the outbreak of the plasmid. So after the finish in these cases, we recommendation for the infection control to implement in the hospital. We have four recommendation and the case about the plasmid outbreak. When the cases is increasing, we should do active screening for the CRA carriers in high risk unit. So we can detect and the hospital one is higher. So this is need to uh, active screening for the patient uh, about the CRA carriage. And second, and also active screening of the patient transferred from the other hospital. So not only in the hospital one, but also when they transfer from the other hospital, so active screening. And then we watching the admission with hospitalization in the previous 12 months on immunocompromised patient. And then contact precaution including isolation or cohorting, yes, this is commonly in the infection control practice, and also hand hygiene measure. So this is important. The risk of further spread in the most uh, affected hospital it means that the hospital one is higher, as large number of the cases were identified from the multiple ward, and the new cases were detected until uh, uh, the last year. And this means that while enhanced infection control measure have been implemented, and the outbreak was still ongoing at the time in this assessment. So don't wait until the end of the outbreak and we're doing that this action, but when we detect in the early of the outbreak, so we should as soon as possible doing activity to decreasing the outbreak. So to take home message, uh, the integration of genomic and epidemiological database and NGS data will be the next frontliner in the IMR epidemiology in order to empower the stakeholder in public health decision. So this is important things. How about Indonesia? If the Indonesia uh, Indonesia, I don't know how deep they doing the measurement of the IMR. But if you want to, if you interest to mastering uh, your skill, it means that to doing the study in Indonesia. So we welcome to you, and please contact to me in this email, and we can do collaboration for the further study. And okay, that is all for the introduction of next generation sequencing to doing IMR. Uh, before this lecture, I want to say thank you very much for the Professor Hiramatsu and also Professor Kirikai and also Professor Hattori to teach me about this field of study. And thank you very much for the Pylori study group. I hope my study group also can establish in Erlangen University. And thank you for give me this opportunity because yes, a long time I'm not talking in the front of audience, so a little nervous on this one. I hope. Thank you very much for this lecture. Otsukare sama deshita. Hi. Thank you.
Hi, Otsukare-sama deshita. Thank you very much, Dr. Firman Setiawan, for very good presentation today. So right now we are getting to the for the discussion session. Okay, Professor Liao, are you still with us?
Yes.
Oh, okay. Thank you to coming to joining us again. So we are having several questions that have been dropped over the Q&A, Q&A in here. So the first one is coming from Putri Amalia. So okay, thank you very much for. I think for this questions, it will be to Professor Liao. Thank you very much for a very insightful presentation. I have a question for you. Is pancreatic cancer is closely related to increasing number of type 2 diabetes cases? Recently, it's also been very interesting, very good, very high. Is it a good advice reducing risk of diabetes mellitus to pancreatic cancer? For example, like we giving advice to reduce the diabetes, it is also correlated to reducing the pancreatic cancer as well.
Okay. Thank you. I think it's a very good question. Yes, diabetes is a risk factor for pancreatic cancer. Previous studies have shown that prolonged diabetes that longer than 5 years, is associated with the increased risk of pancreatic cancer by about twofold. Okay. And in fact, a few years ago, I conducted a study, meta-analysis, looking at studies that studied the association between blood glucose level and pancreatic cancer risk. And the finding was that there was a linear correlation between fasting blood glucose level and the risk of pancreatic cancer. So on average, about every 10 microgram per deciliter increase in fasting blood glucose is associated with a 14% increase of the risk of pancreatic cancer.
Okay. So there is a very close relationship between the diabetes cases with pancreatic cancer. So it is very good advice to reduce or to have a good good lifestyle that reducing the risk of diabetes. So it will also reducing the cancer cases as well. Okay, thank you very much. And also another questions, it is also for Professor Liao. In the recent years, it's from Karina. In the recent years, the microbiota has association with a very wide of diseases, including diabetes, cancer like colon cancer and other cancer as well. Currently, is there any association with the pancreatic cancer? And there is advised when consuming probiotic also good for the gut microbiota. Is it also has a association or just a hypothesis also? What do you think, Professor?
Okay, that's also a very good question. I think for the first question, yes. Emerging evidence suggests that microbiota plays an important role in the carcinogenesis and the progression of pancreatic cancer. I think in the past, it's usually considered, pancreas usually considered to be a sterile organ. But now evidence is clear that gut microbes can gain entry into the pancreas. And the intrapancreatic bacteria can promote pancreatic cancer progression. And also it may be related to treatment effect, because in a very famous paper published in Science, they found that certain bacteria can produce enzymes that can inactivate gemcitabine. Therefore, microbiota might influence the treatment effect associated with gemcitabine. But for the second question is whether probiotics would be helpful in reducing the risk of pancreatic cancer. I think so far we don't have such evidence.
Okay, thank you very much for the answer, Professor. So yeah, microbiota is kind of key for every diseases that develop to our body. So it is good to have a good friends with our microbiota. Okay, so the next question is for Dr. Firman Setiawan. So in your presentation, seems to be the microbial typing using the NGS is very promising to detecting the antimicrobial resistance. I'm a little bit curious whether is it going to replacing the like replacing the AMR detection by the culture-based method than the molecular-based right now? Is it going to replacing or still just um, going together, and the AMR based from the molecular approach will be more advanced that like that?
Thank you very much for the question. That is yeah, it is depend on a situation in the laboratory or the country or the local. So it's mean that for the advanced, I suggest to doing the genomic uh, to detect the IMR or spreading the IMR around the uh, hospital in the hospital on others. It's mean that if uh, we just using the phenotypic or not the conventional method, we cannot uh, precision detection to how they spreading, and this means that not only in the local, also in global. So if we using the genomic approach, we can't compare with other country or something like that. But if we cannot perform this one, so conventional is much powerful. If but we should do this measurement to uh, stopping or to break the chain spreading of AMR. But now, like me in the Chuntendo University, this is open to for you to doing the basic the basic information about AMR in Indonesia. Because like me, I just doing 5 strain of Mursa. We found the different strain, just 5 is different. So it's mean that we have a question mark and in the situation in Indonesia about AMR, and I think this is a good opportunity to us to explore a discovery about AMR in Indonesia using genomic approaching. I think this one.
Okay, thank you very much. So yeah, practically say the conventional method is still useful, but for further more advanced, such as where this resistance bacteria is coming from, so the molecular approach is have the edge
Yeah, yeah.
to detect, and also like the phylogenetic tree and so on and so on. That's
Mm.
very interesting. Okay, so for the next question is for the Professor Liao. So it is from Indah. I am Indah from Brawijaya University. Thank you, Dr. Wei-Chih Liao, for your informative and insightful presentations. I want to ask when we should initiate the screening in patients with hereditary pancreatitis who have a higher risk of early onset of pancreatic cancer, and which test is more promising or effective to detect the disease at its early stage? Thank you.
Okay, I think it's a very important and useful question. I think so far for the general population, there is no guideline to support screening the general public, asymptomatic general public for pancreatic cancer. That is because of the relatively low incidence of pancreatic cancer. But guidelines suggest that in patients with high risk of pancreatic cancer, screening is recommended. And these high-risk groups include patients with known mutations of the genes that increase risk of pancreatic cancer. For example, BRCA1, CA2 mutation patients with family history of pancreatic cancer should be screened. Others include such as Peutz-Jeghers syndrome or like autoimmune hereditary pancreatitis. These certain high-risk patients, they have a high lifetime risk of pancreatic cancer. Therefore, screening every year with MRI and EUS is currently recommended by the guideline. Okay. But I think these are relatively small number of very high-risk patients, and they account for a very small proportion of all pancreatic cancer patients. So most of the pancreatic cancer patients are those without such high-risk factors. So for them, I think it's important to pay attention to the subtle symptoms of pancreatic cancer. For example, new onset diabetes or unexplained weight loss. These are established early symptoms of pancreatic cancer. Thank you.
Okay, thank you very much for the answer. So yeah, a screening using MRI or USG is recommended for those who has hereditary pancreatitis because such a high risk for developing pancreatic cancer in
With
the near
known
future.
gene mutations.
Oh, without no.
Or yeah, for so-called familial pancreatic cancer kindred.
Okay, okay. Okay. So regarding for the screening, is that any like of the age threshold? For example, like more than 40 or more than 50 years old people, it is recommended to perform the MRI and USG for the general populations.
Yeah, I think it's a very good question. And I think that is basically based on expert opinion. The guidelines suggest that such high-risk group should be screened probably starting from either from 50 or 40, depending on the risk factor and whether they smoke or not, or 10 years earlier than the age at which their family pancreatic cancer case occur. For example, if in a patient with familial pancreatic cancer kindred. So if the youngest pancreatic cancer patient in the family is 50 years old, then his family member should be probably screened from 40 years old. But I think this is based mainly on expert opinion.
Okay, thank you very much. So yeah, the age number is it depends on the circumstances of every patients.
Yes.
Okay, yeah, yeah, and also depends on the population as well. Sometimes like maybe in case of like the gastric cancer, Indonesia is very low, like, but in like in China or Japan is very high. So it also depends on the population as well. So okay, for the next question, this is for Dr. Firman. So Dr. Firman, it is interesting lecture for publication purpose. Does NGS data is better than had more chance to be accepted on the publication rather than the only PCR sequence data? How much do you encourage young researchers to master this technique? Okay.
Okay. Okay. Thank you very much for the question. And okay, I was established infection control and prevention research center in Airlangga University in this year. So I hope the future we can recruit young researcher, and also we can and also I will doing a collaborative research with Chuntendo University. So yes, this if if you want to mastering if you're interested about the field study, you can contact to me. We can discuss about this one, and also you can come to Chonta University to a training. Maybe some researcher come to Chonta University like from Malaysia, and also from Iran, from the Nepal, and come to laboratory to learn about and mastering about uh, especially in MRSA. So we are very welcome to do that, and this NGS data is better and had more chance accepted rather than PCR. Sure, PCR is the old method for detection, but when we doing next generation sequencing, we can know the whole genome, and we can explore more than more. It just one strain is enough for you if you have if you're lucky, and you can get what is name specific and a new novel genome in your result is enough. So this is powerful because this is specific and precision measure to doing for the IMR study. I think this one.
Okay. Thank you very much. So yeah, the NGS now is like new trend for any research in this era right now. So yeah, and also even now the third generation of sequencing. So yeah, for example, in my field, the Helicobacter pylori, this previously was the NGS is from the like Illumina base, and then the Pacific Bio, PacBio, it's easy to get the circular DNA of the whole genome. And now also for your informations, it is also developed a new small next generation sequencer called the nanopore from the Oxford yeah, the nanopore. It's cheap. It's small, cheap, but still powerful to have to get a good result, sequence data. So yeah, the young researchers should study about this for the better data in the future. Okay, so next question for Dr. Firman. And this is, thank you for the insightful presentation. I have a question for Dr. Firman. Is there any strategies for active screening of CRA, carbapenem resistant Enterobacteriaceae carriage in setting of high level endemicity, but in the low economic country? Okay, it's a little bit tough questions. Okay.
Yeah. I don't know what happened in Indonesia now, happening in Indonesia now. But if you want using phenotypic methods for detecting the carbapenem resistant, this is more powerful, also powerful and useful for you in Indonesia if you want to doing this one. So this is maybe in the next conference if you want, I can explain how to doing the phenotypic method for the carbapenem resistant, because I told to you that carbapenem resistant not only just one pathogen, but also can spreading the another organism. So it means that detect early detection, not you waiting. You can doing using the PCR or something like that. You can doing the simple method, but it's powerful in your condition, in Indonesian condition. I mean, so but a better you can stock because this is plasmid mediated. It's so easily to uh, delete it in the sorry, deleted in the genome in the bacteria. So you should as soon as possible have to to stock your strain, and we can doing the whole genome sequencing to knowing how to spreading, and this is endogenous or not. Like in my a previous experiment, in just I performed 5 strain, we have we found a large different of the genome, but this is what I'm Morsa. So I mean that if yes, if you want to learn about the CRA and doing using the conventional method, phenotypic method, I think this is better for you to perform in Indonesia. Okay, thank you.
Okay, thank you very much, Dr. Firman. Okay, so now the last question is for Professor Liao. And your last publication is about the AI, artificial intelligence assisted diagnostic test to detect the early stage of the pancreatic cancer. I saw it. It is very promising. Does it come near close for routine clinical practice in the near future? What do you think it's going to be well implemented? For example, like is it going to be 5 years or next 10 years or even closer or a bit loose? And what if this approach for the imaging assisted by AI is combined by molecular testing, for example, the mutation or something that to really increasing the number on sensitivity and specificity to detect the early stages of pancreatic cancer. What do you think, Professor?
Yeah, thank you for the great question. The first question, we are now seeking to obtain approval from the government, because to be able to apply the algorithm widely in clinical practice, we need to have approval from the government. So in Taiwan, we have the Taiwan FDA. So we are now seeking to have approval. So we have to do a clinical study and then apply for approval. After that, then we can apply this in clinical practice. And I hope this will not take too long. I hope it will be less than 5 years, I hope. And my feeling is that it's not for replacing radiologists, it's supplementing radiologists. So I think given the tremendous workload that our radiologists face every day, I think they do need something to help them. And as to whether this can be combined with other modalities, molecular diagnosis, etc., for detecting early pancreatic cancer, I do think this is the right way to go, because it's impossible to make the diagnosis of pancreatic cancer solely based on imaging studies, let alone just based on artificial intelligence. So I think the AI system may help us identify suspicious cases on imaging. And after that, we need to have either use more advanced imaging modalities or liquid biopsy or protein biomarkers, etc., to try to find early pancreatic cancer. Thank you.
Okay, thank you very much. Oh, wow. Now, the condition already submitted for to the Taiwan government to use as one of the method to detect, right, in Taiwan, the AI based.
We are in the process of
Oh,
contacting
okay, in the
the government.
process. So, okay, hopefully less than 5 years, right? So
Okay.
it is very good for the patients because the more we know, in the more early we know the pancreatic cancer cases, the highest chance to be curated or cured for the patients. So
Yes,
okay.
yes.
Thank you
True.
very much for Professor Liao and Dr. Firman Setiawan for very good discussion and very good presentation. We are so honored to listen to from of you. And for the attendees, I am announcing that the attendance link for the workshop is on the chat. Please fill it out, and then usually we will send you the certificate directly for after you fill. But if you still don't get any notification or the certificate from us, so please contact us from our email, and then our host will send you again the email that you can have if you have some queries. And okay, so before we are closing from the chairman for ITT, Professor Inge, and we are going to have a photo session, a little photo session, okay, for Professor Liao and Dr. Firman. Okay, please be ready in maybe in for 3, 2, 1, smile. Okay.
Firman with mask.
Okay. Good. It's good. Okay. So yeah, now it's come to the end. So for the closing, I would like to invite Professor Inge as the chairman of Institute of Tropical Disease to have a closing statement for this whole workshop series. Okay, so Professor Inge, the time is yours.
Yeah, thank you so much, Dr. Langan. Good afternoon, good evening, good morning. Assalamualaikum warahmatullahi wabarakatuh. It's been a pleasure being with all of you since last Friday. First of all, I would like to thank all the wonderful and distinguished speakers in the whole workshop from Friday, 19 March 2021 until this afternoon. Thank you very much for sharing your expertise, updates, and new technological innovations. I really appreciate that everyone has allocated time from each busy personal and professional schedule. This means the world to me, and words cannot describe how grateful I am. It's been an honor to be among such accomplished and enthusiastic participants these 5 days. I realized that a total of 445 participants were involved in all the workshop series, with more than 100 people for each session. I appreciate that there are also overseas participants. We are happy with your enthusiasm and experiments and analysis techniques also in prominent topics updates. Our expectation is participants could get insights on the new trends, up-to-date information, as well as basic technology in gastroenterology researches. Thus, expressing the ideas into research project will complete our duty. Last but not least, allow me to pass my sincere thanks to the organizing committee for the relentless hard work and the enormous effort that made our meeting possible. To all of you, I hope you would have a great day ahead and may we see each other in other occasions. Thank you very much.
Okay, thank you very much, Professor Inge, for the closing speech. Okay, so to all participants, we would like to take a moment completing our satisfactory survey that we sent to the chat box in this workshop. We encourage all the participants to fill the questionnaire, and then the certificate will be sent to your email directly. Once again, thank you very much for joining our workshop series in the last session with the theme of infection and cancer. We do hope you always in good health and wishing you a good luck for the future. Stay healthy and stay safe for everyone. My name is Langgeng Agung Waskito. I am sorry for any inconvenience may occur during the workshop. We hope you to have a great time with us. Thank you very much.
Thank you.
Thank you.
Thank you very much.
Thank you.